Welcome everyone. My name is Adena Collens, and I will be sharing with you some of my recent work exploring the co-evolution and genetic diversity of acquired phototrophy in marine worm Convolutriloba longifissura. So phototrophy is the ability to produce food from light. You may know this about plants, right? And you also may know that corals have acquired phototrophy. They exchange nutrients for carbon from a diversity of photosynthetic microbial symbionts, and when they lose those microbes, you might know that as coral bleaching. But what you may not know is that co-opting phototrophy from photosynthetic microorganisms, including green algae and dinoflagellates, is common across the animal kingdom, shown here in this tree. Every green circle on this tree represents a species hypothesized to be photosymbiotic. However, the white circles here represent where we don't have any molecular data for these organisms, and as of 2018, right? So some of it might have updated, but that, as you can see here, most of the lineages have not been explored, although this is a really cool feature of animal biology and evolution. And so my group of interest is the marine acial worms, are one group with many species observed to have these green photosymbiotic microbes. And actually, the relationships of these worms and their photosynthetic microbes have interested scientists for more than 110 years. Somebody published on these plant animals in 1913 or 12 maybe. But because of the lack of molecular characterization, we still don't know how they primarily obtain, maintain, and pass on their photosynthetic symbionts, although we do have morphology evidence, right? So with that, my study system is Convolutriloba longifissura, shown here under white light on the left, and for scope, what they look like under a petri dish with my hand. They live in shallow marine coral reef environments in nature, but in our study, but you might know them because they reproduce rapidly and asexually. Not only they do sexual reproduction, but you might have seen them because they are also aquarium pests. So you might see worm away in an aquarium store, and that's for these guys. But our sequencing data actually come from Convolutriloba in a aquarium at the Washington, D.C. Smithsonian Natural History Museum with their algae inside of them. And here's a photo of them in our tank for context, again with my hand, just so you can see. Like other acial worms, they eat and host a green algae in the genus Tetraselmis between their cells, not within their cells. Only the algal cells autofluoresce. So the cells you see on this left image are actually autofluorescent so you can see the dispersion of the algae underneath the worm's skin, and it's kind of throughout the full worm other than their eye spots. On the right, we have a light microscope image of the algae with that green arrow. The orange cells are the pigment of the worm. When Convolutriloba are stressed or starving, they actually eat the algae growing inside them, and it's kind of like this internal garden. And so it's not actually known how the Tetraselmis algae molecularly interact with their hosts, or if it's a host-parasite relationship, right, with that gardening. We don't even know what species they are of the algae, or if they show patterns of vertical transmission like the coral or sponge photosynthetic partners. And then this is where our work begins answering these 100-year-old questions. So this is an outline of my pipeline, of my analysis pipeline. I begin with non-targeted, low-coverage whole genome sequencing DNA. The reason we want non-targeted sequencing, it allows us to capture more genes and non-coding regions for organelle assembly and to create more robust evolutionary trees than single targeted gene sequencing approaches, which make up the vast majority of existing sequence data for Tetraselmis. Then I created this custom pipeline to do quality filtering, de novo assembly, annotation, alignment, and building gene trees to compare our Tetraselmis gene sequences against existing Tetraselmis data in GenBank. My current output are draft assemblies of the mitochondria and from the worm and the algae, and also the algal chloroplast. And I've also made several marker gene trees from these assemblies and from the nuclear DNA, which is not part of these assemblies. And I will highlight subsets from those gene trees today, because this is a 6-minute talk. The vast majority of acial symbiont Tetraselmis identification has actually only been done with small subunit rRNA. This is a universally conserved gene used to identify microbes that we can't culture in the lab. This here is a SSU RNA tree of tetraselmis that are extracted from environmental worms. And so actually, so with this green arrow, I'm pointing to our tetraselmis, which are most similar to sequences from other tetraselmis that were associated with these Convolutidae worms. And they are within this large cluster of sequences of SSU that are associated from Convolutidae worms, which is exciting. But that said, the SSU RNA gene, although it's overrepresented in the database, it is a very slow evolving gene. It's highly conserved, it's slowly evolving, and so that limits its usefulness for us to resolve the species of this algae. And so we obviously need to use additional genes, which we have. This here is a subtree of the RBCL, also known as Rubisco for any plant folks in the room, which is used for plant taxonomy as it is from the chloroplast of the algae. The green arrow here shows our Convolutriloba tetraselmis is closely related to other tetraselmis from acial worm hosts, such as Simsagitifera, also up there. These are represented on this tree by the orange squares. However, many of the closely related sequences for RBCL are also from free-living tetraselmis, which indicates their host association may not actually dictate as strongly as other vertically inherited photosymbionts, like such as those in coralline sponges. So it's a more complicated question because they, from morphology, we know that they inherit their algae from asexual reproduction, which is most of the time, but they are free of algae during sexual reproduction. So this is some of the parts of what we're trying to find out here. So in conclusion, the main takeaway here also is that we have these organellar genome assemblies that are the first to molecularly characterize the Convolutriloba associated tetraselmis. And next steps, our further work is to answer these central questions. Who are the tetraselmis and how do they co-evolve with their hosts? This steps include completing and polishing the organellar genomes that we've assembled and resolving the evolutionary history using multi-gene tree approaches and identifying them with co-evolution analyses. With that, I want to thank my advisors, Mihai Pop and Alan Collins. I also want to thank the Rita Colwell Travel Fellowship from University of Maryland for allowing me to be here with you. And with that, I would love to connect and take any questions. Thank you.
We have a couple of minutes for questions.
Thank you. Very interesting topic. You show example of symbiosis with algae.
Yes.
I wonder if there are examples of symbiosis with prokaryotes.
Yeah, so actually on the original animal tree that I was showing, these actually include, I would point you to this paper actually, if you're interested. I will go back to that first. So if you read this Melo et al. Paper, some of these are non-eukaryotic photosynthetic organisms, including cyanobacteria. So cyanobacteria are the prokaryotic photosynthesizers that you might be interested in. But yeah, I would encourage you to read the paper. It's great.